So we are going to be starting with genetics and biotechnology. This is going to be the last topic of this module. So for the rest of our classes, let's see 2 or maybe 3, how the course will go. We are going to be studying only this topic. So let's start by understanding what this is. When we were studying the cell, we spoke about this term genetic material. So what is genetic material? What comes under genetic material? DNA, yes. Remember DNA? We used this term. So DNA is the genetic material which we have, and we are going to study more about it here. What does this genetic material do? Any idea about that? Yes. So parental traits are transferred to the offspring. That's the job of your genetic material. How some of your features are like that of your mother and some of the features are like that of your father is because you have received DNA from them. This DNA goes from one generation to the next. It's basically how information would be traveling. Now this whole study of this genetic material, which we are saying is DNA, for some organisms it can be RNA also. Their study and how they are going to be inherited. That whole process, when we talk about studying that, we use the word genetics for it. Okay, so please write this term genetics. Genetics is the study of genetic material and its pattern of inheritance. Genetic material and its pattern of inheritance. Now we also had to see this question. So just going slightly backwards. Actually, this I think I've copied it wrong. This should be 5. Okay. So the endocrine system which we studied, this is a question based on that. D. Okay. So you can just look back and see what these hormones are about. The pituitary gland, by virtue of its trophic hormones, controls the secretory activity of other endocrine glands. Which one of the following endocrine glands can function independent of the pituitary gland? Now, if the question was just this much, then also it would have been a complete question. Okay. But they've given you one more line, and that is just to give you some additional information and maybe, make the question appear a little bit more technical. So the first statement is saying pituitary gland, by virtue of its trophic hormone, controls the secretory activity of other endocrine glands. ये बंद करा दीजिए ये वाला भैया, ये वाला बंद करना. Now this is actually the definition of the term trophic hormone. Trophic hormone is that hormone which is released from one endocrine gland and it acts on another endocrine gland. एक endocrine gland से release हो रहा है, लेकिन उसका impact is coming on another endocrine gland. Such a hormone is going to be called a trophic hormone. So this is saying pituitary gland has trophic hormones, and which one of the following endocrine glands are having trophic hormones for them? That's what you need to find out. So do you have a hormone for thyroid for thyroid which is coming from the pituitary gland? Yes. So we have TSH for gonads, LH. LH is the hormone which is stimulating the gonads to produce the sex hormones. Okay. For adrenals, ACTH. और इसके तो नाम में ही TH है. Right? Now you can understand this name better. Adrenocorticotrophic hormone. Adrenocortico, because we had learned yesterday कि this is supposed to be stimulating the adrenal cortex. ठीक है, तो adrenal cortex को stimulate कर रहा है, तो adrenocortico, और because it's stimulating another endocrine gland, it is a trophic hormone. For parathyroid, we don't have any such hormone, so the answer is going to be parathyroid. ठीक है, okay. Coming again to genetics. Now we have said that there is a genetic material and we are studying that, that is genetics. So we'll go further into seeing what is going to be there in the genetic material. This DNA and RNA, the Na part is nucleic acid. So these are two types of nucleic acids which we have in nature. So all genetic material of all living organisms is made up of nucleic acids. Okay, this is what is responsible for transmission of characters. If you look at DNA, DNA का full form है deoxyribose nucleic acid. Okay. So we'll talk about nucleic acids now. Please write down nucleic acids. Nucleic acid form the genetic material of all life forms. Nucleic acids form the genetic material of all life forms. They are responsible for transmission of characters from one generation to the next. Transmission of characters from one generation to the next. It is made up of please write what it is made up of. Phosphate group. It is made up of phosphate group, a pentose sugar, pentose sugar means this is a carbohydrate. Sugar is to represent that it is a carbohydrate, and pentose means there are 5 carbon atoms. Basically, this is a monosaccharide. That was the first thing we had learned about when we started studying biology. So this is a monosaccharide with 5 carbon atoms, and you have 2 of these in nature. You have 2 types of pentose sugar. One is called deoxyribose and the second is called ribose. It is basically the difference in these sugars that is creating DNA and RNA. Deoxyribose and ribose. The third thing present in nucleic acids is nitrogenous bases. Nitrogenous bases further are of two types. You have purines, and there are two purines in nature, adenine and guanine, and pyrimidines. You have three pyrimidines: uracil, cytosine, and thymine. Now, you don't need to know the chemical structures of any of these things, but you need to know what they are. Nucleic acid or genetic material of all living organisms will always have three things. This is fixed phosphate group, pentose sugar, nitrogenous base. In phosphate group, there is no variation. In pentose sugar, you can have deoxyribose and ribose sugar. Nitrogenous bases, you have two types, purines and pyrimidines. Okay. Now we are going to go and talk about the two types of nucleic acids, which are your DNA and RNA. So let's see what's the difference there. So you can write this as types of nucleic acid. You have two types of nucleic acids, DNA and RNA, and you can write their full form also. Deoxyribose nucleic acid and ribose nucleic acid. If we look at their features, what we find is the first point of difference will be in the type of sugar. You have deoxyribose in DNA and ribose sugar in RNA. They are both pentose sugars, but the sugar which they have, that is different. When it comes to nitrogenous bases now, they both have both the purines, that is adenine and guanine. They both have pyrimidine, cytosine also. But the last nitrogenous base, that is variable. Between DNA and RNA, what we have is thymine is present in DNA, and in place of this thymine, you have uracil. So the structures of both DNA and RNA have two purines and two pyrimidines. But pyrimidines have a point of difference, thymine in DNA and uracil in RNA. From here on, we are going to be referring to these with standard abbreviations A, G, C, T and U. This is fixed everywhere, A, G, C, T, U. Now the whole arrangement here, the structure, if we talk about, we find that the structure of RNA is single-stranded. Single-stranded means there is one chain, there is one structure, one ribbon-like thing, one strand. RNA is single-stranded, but when it comes to DNA, actually no need to write this term. We still have to come to this term double helix. We'll understand what this is. Here you can simply write DNA is double stranded. Being double stranded means two chains, two ribbons, two strands. So DNA is double stranded, RNA is single stranded, and the sugar is deoxyribose in DNA, ribose in RNA, and thymine in DNA, uracil in RNA. The phosphate group remains the same. The next thing which we are going to go into is the structure of DNA. How all these things are arranged in DNA, what is the logic of a double strand. We'll see all of that. See, when it comes to genetics, understanding each and every part is very important. This is like a puzzle in the sense that if you miss one piece, you'll find it very difficult to, understand what's happening. So here, you have to make sure that every bit you are understanding, because the next one is going to be based on that. Please ask if you are not understanding any point anywhere. So if we look at the structure of DNA, we say we have a double-stranded structure. So these are the two strands. When we examine them, we find that the base of these two strands is made in sugar and phosphate. So we call this the sugar-phosphate backbone. The two strands, their base is made up of the sugar and phosphate, and in between this, you will have nitrogenous bases being present. Now, there are 4 nitrogenous bases in DNA, and they can be present in any random sequence. Okay, there is no rule as such कि purine आना चाहिए पहले pyrimidine आना चाहिए. You have 4 bases, they can be arranged in any manner. And if we talk about the whole DNA of a person, the sequence of these nitrogenous bases, that is unique to every person. Okay, we can have like thousands and thousands of these base pair bases, like I'll write any random sequence. T, C, G, A, C, G, A. ऐसे कुछ भी हो सकता है, and think of like if you have such a large number of this sequence, so many combinations are going to be possible, and this combination is what makes each of us unique. Now, these nitrogenous bases have a property called complementary base pairing, in which the nitrogenous bases form bonds. Purines join to pyrimidines, and which purine joins to which pyrimidine, that is fixed in nature. Adenine will join only to thymine. There is a double bond between them. And guanine will only join to cytosine, and this happens through a triple bond. This relationship is fixed. It's complementary, and it's always going to be like that. Now, because of this, if you know what is present on one strand, you will easily be able to predict what is going to be there on the second strand. What will come up opposite to T? C, G, G, C, A, T, A, T again, C, G, C, T. This is the logic of complementarity. You know what's there on one strand, you'll know what's there on the second strand also. So these nitrogenous bases, they will be forming bonds between themselves, and these bonds are going to make sure that these two strands stay together. Okay, it'll make sure that the two strands are, going to be joined together. This whole structure ultimately will twist on itself. Let's see how that works. Then we'll write down a little bit about this. So this is that same image. They've shown two strands. The sugar phosphate backbone is making those strands. In between, you have the nitrogenous bases forming bonds. Adenine with thymine, guanine with cytosine. Okay. And the whole thing, we've drawn this as parallel lines, but the whole thing will twist on itself. Okay, so this is the twist which is there. And this gives the actual shape of DNA. And the bonds here, they kind of give an appearance of a staircase-like structure. So the twisted staircase-like structure is what DNA is going to be. And this we are calling the double helix DNA. The term which was used in the last image, that's what is created. After that twist, we say this is double helix structure. This structure of DNA is also a very famous Nobel Prize winning discovery. So we'll write about this. We'll write about the structure of DNA. Structure of DNA. The two strands of DNA, are formed of a sugar and phosphate backbone, The nitrogenous bases present on these strands pair up using complementary base pairing, where adenine joins to thymine. Adenine joins to thymine through a double bond, and guanine joins to cytosine through a triple bond. The two strands then twist together. Into a spiral structure called the double helix DNA. Okay. So now that we know about the structure of DNA, we're going to put this whole thing in alignment with the concept of the cell, which we already know. So do you remember the cell? We studied that 1 million years ago. So when we studied about the cell, we said the cell has a nucleus. Every cell has a nucleus, and the nucleus is the location for the genetic material. All the genetic material of the cell is present within the nucleus. Further, we said that this genetic material is linear. It is in the form of rod-like structures called chromosomes. Chromosomes are rod-like structures made up of DNA. So DNA is the main genetic material of the cell. It is arranged in the form of rod-like structures called chromosomes, which are present in the nucleus of every cell. So every cell in your body has genetic material present in its nucleus. It is present in the form of chromosomes and made up of DNA. So your basic genetic material is the DNA. Okay. Till here we've already mentioned, and this is now a new concept which we are going to introduce. A gene, which is a segment of DNA. Now, what we have said or what we have established till here is that passing of information from one generation to the other is happening through genetic material. It's happening through DNA. That means all your features, they are working according to the directions of the DNA. आपके बाल कैसे होंगे, आपकी आईज कैसे होंगी, आपका पैनक्रियास कैसे काम करेगा, all of that information is present within the DNA. And that is actually what makes DNA such a large molecule. Because so many things it needs to, so much information it needs to store. Right from how your organs will work, how your cells will divide, how your brain will work, how your memory will work, how your eyes will look like, what kind of diseases you'll have, all of that. So it becomes a large, bulky molecule. Within this whole DNA, what happens is that smaller areas take the responsibility of some parts of the information. जैसे कि ये अगर एक DNA का part है, so let's suppose this much part, one segment, one small section of DNA, this has the information for the color of your hair. ठीक है, पूरा अब इतना बड़ा है तो हर छोटा छोटा पार्ट छोटी छोटी इंफॉर्मेशन of one particular character is going to keep. Say the next part, the next segment that has the information for the texture of your hair. Okay, like this we'll have segments dedicated to individual traits, and these segments can be called genes. So we can say this part is the gene for hair color. The next part, the next segment is the gene for hair texture. इस तरह से पूरे DNA पे सब features का genes are going to be present. Okay. Now the next important question is that we are saying that DNA has information, okay, or this gene will have information for hair color or eye color or any such feature. How is the hair color actually going to get formed? ये तो information है. Hair color बनेगा कैसे? How will we see the impact of that gene? For that, you need production of proteins. Whatever information is there with each of these genes, that is going to be converted into proteins. And it is only after the production of proteins in your body that we'll see the impact of this information. So the gene will show its impact by producing proteins. Ultimately, in your body, all the features which are there, they are going to be expressed or they are going to be seen based on what proteins you have and what proteins you don't have. So genes में या DNA में information है. To get the impact of that information, proteins have to be produced based on that information. Only then that feature is going to be expressed. ठीक है? Okay. Now the next thing which I want to ask you, if you can recall, where are proteins produced in the cell? Ribosomes. So we had structures in the cell which were called ribosomes, and they would be producing the proteins. Where are they found? Okay, they can be found attached to endoplasmic reticulum or in the cytoplasm freely. Basically both these things are in the cytoplasm only. So ये है ribosomes present in the cytoplasm. This is your cell. This is the nucleus. We are talking about how the gene is going to show its impact. Where is the gene present? Which part of the cell? Nucleus, right? We've just seen that. DNA, genetic material, it's all present in the nucleus. So, ये है DNA, इसका segment को हम बोलेंगे gene. Now, the information is in the nucleus, proteins बनेंगी cytoplasm में. That means somehow this information from the nucleus has to go to the cytoplasm. Now DNA, very huge molecule, very large molecule, it cannot come out of the nucleus to give the information to the ribosomes. So what it does is, it creates a messenger. The gene or the segment of DNA which has to show its impact, that particular information is given to a messenger called messenger RNA or mRNA. सिर्फ उस जीन के लिए एक छोटा सा mRNA बन जाएगा, and this is now capable of coming out of the nucleus. This comes out of the nucleus, goes to the ribosomes and tell the ribosomes that this is the information I have brought. Based on this, please prepare the protein. Think of this like a recipe. कि आपकी eyes का color बनाने के लिए whatever needs to be done, that has to happen through proteins. Okay, so a specific protein has to be made for your unique eye color, whatever that is. The information is with the DNA segment. That segment gives its information or gives its recipe to mRNA. ठीक है, कि ये रेसिपी है, कैसे प्रोटीन बनानी है, मेरे पास है, आप ले लो. आप न्यूक्लियस से बाहर जाओ, go to the ribosomes and tell the ribosomes that okay, this is the recipe, you read this recipe and based on that you prepare a protein. एक बार वो प्रोटीन बन जाएगा, then your eye color is going to be formed. ठीक है? There are two steps here, DNA से RNA, RNA से proteins. We have technical terms to describe these steps. DNA to RNA, it's called transcription. RNA to proteins, it's called translation. The whole idea here, that is called gene expression, and we are going to be writing about that. Please give the heading gene expression. Segments of DNA tell individual cells, segments of DNA tell individual cells how to produce specific proteins. How to produce specific proteins. These segments are called genes. These segments are called genes. It is the presence or absence of specific proteins that gives an organism its characteristics. It is the presence or absence of specific proteins that gives an organism its characteristics. Transcription of the DNA segment of gene, transcription of the DNA segment or gene into messenger RNA, takes place in the nucleus of the cell, and translation of messenger RNA into specific proteins takes place in the ribosomes present in the cytoplasm. Is this fine? Okay. Now we are going to be applying this for making vaccines. So we have studied a number of vaccines. This is going to be one more method we are going to be adding to how vaccines can be made. So we'll just review the process of making vaccines. Vaccine making requires coming of an antigen. So we discussed the concept of antigen antibody yesterday also. So for vaccine making, what happens is when an antigen comes, B cells recognize it, B cells produced antibody against it. At the same time, immunological memory is created for that particular antigen. So B cells have this property that they will remember the antigen, and if this antigen comes at any point again, it is going to produce, B cells are going to produce such a large number of antibodies that that antigen will be immediately destroyed and will not cause the disease. We studied different ways in which we can deliver this antigen. We studied about giving a live and weakened organism. We studied about giving a killed organism. And we also considered that there is a possibility that we can just give some subunits. You take a pathogen, you study its structure, and suppose you find out that for a particular pathogen, its proteins are acting as an antigen. So what we could do is, we can just take this protein instead of that complete live or dead organism and only give the proteins. अगर B cells का काम सिर्फ protein recognize करने से चल रहा है, then we can just give the protein and we call this a subunit vaccine. Now, we are going to be making nucleic acid vaccines. न्यूक्लिक एसिड वैक्सीन्स मीन्स कि आप या तो डीएनए यूज कर रहे हो या आरएनए यूज कर रहे हो टू बी डिलीवर्ड फॉर मेकिंग अ वैक्सीन. Basically, what you are going to be doing is you are just using this process. Okay, we know there is a natural mechanism in which proteins are produced. So एक तरीका तो यही है कि आप प्रोटीन दे दो सीधा. You're calling that the subunit vaccine. If instead of the protein, if you find out which mRNA is producing that protein, you can give that mRNA. This mRNA, once given as a vaccine, it will go into the body, it will produce proteins. Once the protein will be produced, B cells will be recognizing it as the antigen, and the immune response and the memory which you want to be created, that is going to be created. Just adding one more step. Okay. What is the urgency to comb your hair? I really want to know this. I can't help but concentrate. मैं बहुत देर से सोच रही थी, मैं नहीं देखती हूँ, but sometimes it's like something is too strange for everybody else also not to know, you know. So everyone should know that you can use your cell phone and comb your hair in the class. This is a site I really can't like delete the image from my mind. Okay, coming back to mRNA. So if you give mRNA, that becomes an mRNA vaccine. ठीक है? अगर further if you want to go, you will have a DNA segment which is responsible for producing the mRNA. So mRNA देने की जगह you add one more step to it. mRNA दिया या DNA दिया. You give the DNA, DNA will make mRNA, then mRNA will make proteins. Just adding a few more steps. But ultimately, once the protein is produced, it will act as an antigen. B cell will recognize it, produce antibodies, and create memory. Till the time you have memory, you are protected against a particular pathogen, a particular disease. ठीक है? So इसमें अब क्या होगा कि या तो प्रोटीन दे दो या mRNA दे दो. If you give mRNA, you say this is RNA vaccine. Then you can give the DNA segment or the gene which is responsible for making this protein ultimately, and when you do that, it becomes DNA vaccine. Concept remains the same, immune response, everything remains the same. Just this is an alternative form of delivery. For COVID-19, we were able to create both these types of vaccines, and those examples are there with us. RNA in particular, this was something which was being researched for many years, but now this has successfully come up as a vaccine. One of the most successful vaccines for COVID-19 was the Pfizer and Moderna vaccine. These are two companies which are making COVID-19 vaccines. And these RNA vaccines, actually अभी तो debate भी चल रहा है. Both of these say हमने पहले बनाया, so that controversy is also going on. But mRNA vaccine has received a Nobel Prize this year. Okay, so this is important in that sense also. For DNA, you have an indigenous company made in India vaccine. You have Zydus Cadila making Zycov-D. This is also for COVID-19. और इंडिया ने एक mRNA वैक्सीन भी बनाया which is Gemcovac. This they had made for Omicron virus. So all of these are COVID-19 vaccines. We'll write about this. Please write DNA vaccine. DNA vaccine. Direct introduction of the DNA sequence coding for the disease specific antigen. coding for the disease-specific antigen. The production of target antigen takes place in the body of the vaccinated person. Okay, and make sure you write the example zycov D. सी आपकी बॉडी को या बी सेल्स को कोई लेना देना नहीं है कि ये एंटीजन वायरस की बॉडी में बन के आ रहा है या आपकी बॉडी में बन रहा है. As long as it is that antigen which it recognizes as the COVID-19 antigen, if it's that same protein to which it is responding, it will have the same response and the same protection against the pathogen, in this case COVID-19, irrespective of where the antigen was produced. So even if you are delivering this DNA or RNA, the result is going to be the same. B cells respond only to the antigen once it is produced, irrespective of where it is produced. Okay. Please write RNA vaccine. RNA vaccine. It is introduction of the mRNA sequence coding for the disease specific antigen. Coding for the disease specific antigens. Okay. And you can write examples: Pfizer and Moderna vaccine and Gemcovac made in India for Omicron virus. नहीं, एक बार ये प्रोड्यूस हो जाएगा उसके बाद रिस्पॉन्स सेम है. The only thing why we are trying to find these alternative methods is because we have to ensure that we don't give the disease. That's one main consideration of the vaccine making process. So when you are giving organisms in live or dead form, you still have some higher chance of getting a response. इस case में वो disease causing element बहुत ही almost it cannot be there. And the other advantage is this is easier to extract also for us. The problem why it couldn't be done for so many years was the delivery. RNA especially, it's easier to find the RNA segment, but it was very unstable. So you couldn't really create a vaccines for many years, but now we have done this successfully. Okay. All right. Okay. Now, if you look at this, we have said that for a gene to have its impact, you need proteins. Think of a situation, a technology where we don't want a gene to express itself. Suppose there is a gene whose defect which has become defective, and now it is going to create some problem. It is, say, causing some disease. Okay. In this case, we don't want the gene to express. We don't want to see any impact of the gene. How we can do this is, we can find a way to avoid or stop production of this protein. ठीक है? अगर protein production नहीं होगा, there will be no impact of the gene. So the way to stop it would be, we have to do something to either of these steps. ठीक है? या तो आप DNA से RNA ही बनने मत दो. ठीक है? और अगर RNA बन जाए, then you find a way so that RNA does not change or to proteins. Okay. When you do that, we say we have silenced a gene. So this is a natural mechanism of the body which we have now developed into a technology called gene silencing technology. So ये naturally होता है, but now with its use, we have been able to apply this to various situations. Like you have some defective gene, like you identify कि एक gene है जिसके वजह से cancer हो रहा है लोगों में, ठीक है? Now what you can do is you can apply gene silencing to that gene. If that gene will not express itself, the impact of that gene will not be seen. Okay. In other organisms like okay, we spoke about microbial resistance. Okay, so we find out that there are some pathogens which are now resistant to the medicine, and we study them and we find that resistance is happening because of some special gene which they have. Okay, if we can silence that gene, the impact of that gene will not be seen and the pathogen will no longer be resistant. फिर वो दवाई से मर जाएगा. So we can choose to silence genes, and this technology has been in place for many years now. Of course, its use is not very wide, but we see a lot of potential of using this. अभी इसको resistance के cases में, जैसे pest resistance हो रहा है plants में, in those cases we have been trying to use this. This is also a Nobel Prize winning technology. Okay, just write one line, then I'll tell you a little bit more about how this technology works. Gene silencing is interruption or suppression of the expression of a gene at transcriptional or translational level. At transcriptional or translational levels. Silence means destroying the gene. Parul is asking. No, Parul, it doesn't mean destroying the gene. आप gene को बिल्कुल कुछ damage नहीं करने वाले हो. You are just going to stop the next two steps which are going to happen. Now, the Nobel Prize winning technology was a post-transcriptional technology. Post-transcriptional here means कि DNA है, DNA से mRNA बन गया. That means transcription has taken place and now you're going to do some intervention. This intervention is done through presence of a special type of RNA called iRNA. This iRNA, or its full name is RNA interference. The special feature of this RNA interference is that this is an exception to the normal RNAs which we have, because this RNA is double-stranded. We've studied that RNA is supposed to be single-stranded, right? But ये एक exceptional RNA है जो double-stranded होता है. We have this RNA interference or iRNA in our body also for checking different genes. But now in this technology, we can create RNA interference based on whatever gene we want to silence. ये iRNA mRNA को attack करेगा and mRNA then will not be able to make proteins. So when no proteins are formed, the impact of this gene is not going to be there. Gene है लेकिन उसका कोई impact नहीं है, so gene silent है. ठीक है? Write down one more line. Post-transcriptional gene silencing. post-transcriptional gene silencing inhibits protein synthesis inhibits protein synthesis in target cells using double-stranded RNA interference or iRNA. This iRNA attacks the mRNA created during transcription the mRNA created during transcription so that no proteins are formed. Now, I'll give you one example also of this technology's use. Of course, you have all those examples of having potential uses in cancer and other diseases and resistance. But we also have the possibility of creating products with some favorable properties. जैसे कि we have produced a special variety of tomato. Now see, with tomato, the problem is that tomato has a very short shelf life. So farm से market तक आते आते बहुत सारे टमाटर वैसे ही खराब हो जाते हैं, ठीक है? So their ripening process is a little faster than what we would like to make sure that they can stay fresh for longer. So what was done here is using this gene silencing, जो टमेटो की फास्ट राइपनिंग प्रॉपर्टी है, that gene was silenced. And when it did not ripen quickly, or when that ripening was delayed, the tomato could now have a higher shelf life. It could stay without getting spoiled for a longer time period. और ये टमेटो वैरायटी को बोलते हैं फलवर सवर tomato. Okay, so this is an example you can write for gene silencing. Okay, now see, any such kind of application, you know, where you're using genetic material from living organisms to create something useful, the term we use for that is basically what biotechnology is. Technology, wherever we are going to talk about this word technology, it means something which you have created for the betterment of human life. That's the objective of technology. Having bio with this means that we are using living systems. Okay, so wherever you use living organisms or anything from living organisms to create something useful, that becomes biotechnology. Is it an example of genetically modified crops? Yes, we'll talk about what are genetically modified crops a little later. But because we have changed how genetic material was working, this would be a genetically modified crop. We also use the term genetic engineering here. It's a broader term. So genetic engineering means simply that you are altering the genetic material in any way. जैसे हमने जीन को काम नहीं करने दिया, that becomes alteration, that is genetic engineering. Can we temper DNA to create better offsprings? Yes, we can do that. We'll learn about that also. We learn about how we can change genetic material, how we can bring different form of modifications. Okay, just write this term, biotechnology. It is the area of biology. It is the area of biology that uses living organisms, their processes, systems to manufacture products or technology intended to improve the quality of human life. Intended to improve the quality of human life. It uses genetic engineering. Which is a process of alteration of genetic material. Okay. Now we have to talk about one special organism when we are talking about how genes are working. In continuation of that, we have to talk about the viruses which have a slightly different method of gene expression sometimes. For viruses, what we see is that they have two types of genetic material. You can have some viruses who have DNA as their genetic material, just like all the other organisms, just like we have. But we can also have viruses with RNA as their genetic material. When I say RNA is their genetic material, what I mean to say is that their genes, the genes which are having the information for how an organism is going to be working, that gene is in the form of RNA. That is an exceptional thing which can be there only in a virus. बाकी जितने organism है, bacteria, fungus, amoeba, plants, animals, all of them, they have DNA. Their genes are formed of DNA. But virus is that exceptional organism जिसमें ये RNA भी हो सकता है. Their genes, their genetic information can be in the form of RNA. Because of this, we have two types of viruses, DNA virus and RNA virus. Single strand DNA found, yes. जैसे double stranded RNA हो सकता है as an exceptional case. सिंगल स्ट्रैंडेड डीएनए भी हो सकता है. दैट इज possible, though very rare, but it is possible. Coming back to virus, so we'll talk about examples in each of these cases. So DNA virus में we have adenovirus. This is a very common virus variety and there are many types of adenovirus which cause many diseases. जैसे कि common cold इससे होता है. Yeah, you recently had the outbreak of your eye flu or conjunctivitis. That could be because of one variety of adenovirus. And there are many other adenoviruses which actually have no impact also. Neutral type of adenovirus is also no human diseases being caused. So this is a very broad category in DNA viruses. Then one more example I'll give you. Chickenpox virus. Now when the DNA, when the virus is going to be a DNA virus, its gene expression, the genes are in the form of DNA, so gene expression will be exactly like what we have learned. DNA होगा, ट्रांसक्रिप्शन होगा, RNA बनेगा, then translation, and finally gene expression through formation of proteins. Okay, this is the process which you've already learned. Now let's see what's happening with RNA. RNA में further there are two types. One is the normal RNA or it is called non-retrovirus. This has RNA as its genetic material. अच्छा पहले example बताती हूँ. Here you have your COVID-19 virus or the dengue virus. We spoke about the dengue virus yesterday. They have RNA as their genetic material and what happens is RNA है तो RNA से सीधा प्रोटीन बन जाएगा. No other step is needed. Okay. But there is another kind of virus which is called a retrovirus. Here the characteristic example is HIV. And what this virus does is, of course it is an RNA virus, so its genetic material is in the form of RNA, but it forms DNA. In a normal case, DNA is supposed to form RNA. But यहाँ पे exceptionally what is happening is RNA is changing to DNA. This is the ulta process, so we are calling this reverse transcription. The word retro also means going backwards. So if you talk about like retro fashion or retro music, maybe you're talking about the music of the 80s and 70s like that. That's what is retro. So इसमें retro यही है कि DNA से RNA बनता है, but यहाँ पे RNA से DNA बनेगा. And this is the special case of HIV. Which disease does HIV cause? AIDS. Okay, so the AIDS causing HIV virus is a retrovirus. So, to summarize this, all living organisms on the planet, they have DNA as their genetic material. But viruses can also have RNA. Some viruses which have DNA as their genetic material, they are the DNA viruses, follow the same process of gene expression. RNA viruses can be of two types, non-retroviruses, retroviruses. Non-retro जो RNA से प्रोटीन बनाते हैं and retro RNA से DNA बनाए. ठीक है, और डीएनए के बाद वाला प्रोसेस फिर से वही सेम होगा, डीएनए फिर आरएनए फिर प्रोटीन, लाइक दैट. ठीक है, इज दिस फाइन? ओके. Another small point which I want to make here is, see, when organisms are reproducing or when they have to form new cells, what they have to do is they have to create new DNA also. Okay, so DNA or genetic material will have to be copied, will have to be replicated. So DNA replication would mean creating new DNA or creating copies of that DNA when it is supposed to be transferred to any new cells. Now, this DNA replication now we can do in the lab also. We can create DNA artificially, all of that. So we've done a lot of extensive study also on how this works. There is this process of proofreading which happens when DNA replicates itself. When I say proofreading, that means there is going to be a process to check how the copy is being made. जैसे कि इतना DNA है, if you have to make a copy, the sequence has to be exactly this. Because one base, if it changes, if it goes out of place, the feature will become something else. The property will become something else. ये formula बिल्कुल exactly follow होना चाहिए. Even a slightest change in this recipe is what you call mutation or error. If that comes, that will change whatever it is supposed to express. So the cell, whenever DNA is replicating, whenever a new DNA is being formed, makes it very sure कि ये properly check करे कि T के बाद C ही आया, C के बाद G आया, G के बाद A आया. Because if this is not so, something else is going to be created. Okay, so DNA replication में proofreading is an integral process. But when it comes to RNA, if you talk about new RNA being created or RNA replication happening, there you have no proofreading. RNA is a simpler molecules. Its replication is also simple. And because there is no proofreading here, the chances of error are very, very high. So while creating new RNA in RNA organisms, there will be errors. And that is why you will find when viruses are RNA viruses, very quickly they will have new varieties. And I think we've all observed that with COVID. ठीक है ना, कोविड में बहुत जल्दी जल्दी न्यू वेरिएंट्स आ रहे थे, अल्फा, बीटा, डेल्टा, ओमिक्रॉन, और अभी तक आते जा रहे हैं, राइट? Why that's happening is because it's an RNA virus and there is no process of proofreading. So उसमें जो bases हैं उनका sequence, उनका कुछ भी जो है change होता है और उससे virus की properties change हो जाती हैं, and that's how it becomes a new variant. ठीक है, right? Okay. Now when we study this whole thing, these sequences, we use a term which is called genome. I'm sure this term you've heard. ये बहुत current affairs में आती हैं चीजें. So genome and genome sequencing. Genome now is an organism's complete genetic material. If I'm talking about one person or say one virus, and I'm talking about what is his complete DNA like. If I'm referring to the complete DNA, that means all the chromosomes with all the genes and everything. The word for that is genome. Okay. I can also use this term for the whole species. Like I can say human genome or COVID-19 genome. That will tell us कि COVID-19 species में जितने viruses हैं, उनमें जितने genes हैं, उन सब का total information, that is what is going to be collectively called a genome. Okay. If we talk about writing their sequence, जैसे the sequence which we just saw, like any sequence which is there, sequence of nitrogenous bases basically. If you take the genome of a person and from one end to the other, actually it's not in ends, it's supposed to be in parts. So if all the parts you start writing कि हाँ पहले A आ रहा है, T आ रहा है, G आ रहा है, C आ रहा है, and then along with this you will also write कि A, T, G is for brown hair color. Then whatever is the next sequence you write, this is for green eyes. आप सीक्वेंस लिखोगे बेसिस का, आप लिखोगे कौन सी जीन है एंड what how it is expressing in terms of feature, how it is working, how it is functioning, all of that. This whole information, just gathering this whole detail of base pair sequences, that is genome sequencing. And if you do that for the whole species, then it becomes like COVID-19 genome sequencing, bacterial genome sequencing, or human genome sequencing, whatever you are doing. Can you think of how this can be used? या ये क्यों करना है? Because this is a very, very difficult process. You have thousands and thousands of these base pairs. There's so much data which is collected. इसको calculate करने के लिए, assemble करने के लिए, you need supercomputers. So it's a very, very tedious process. But this is something which has been of interest for many years now. Like 1970s से human genome sequencing projects start with. Why do you think we need to do this? Disease identify कर सकते हैं, ठीक है? If you have the information of what are the different genes and what is their sequence, we can identify some parts of the genome which can be linked to some diseases. ठीक है? जैसे कि okay, suppose there is this gene which is responsible for causing lung cancer, or people who have that particular sequence, they are at a higher risk of having lung cancer. So in that case, what can be done is that we can start checking people for those genes. और जिनमें आएगा उनको पहले ही बताया जा सकता है कि तुम तो स्मोकिंग बिल्कुल अफोर्ड नहीं कर सकते हो, राइट? या और whatever other trigger factors are there. So we can learn how diseases are being formed, especially when there is a genetic nature involved here, right? Identifying the specific to identifying ही है ये, the specific create. Okay, so if you are saying you to identify a specific creature or identifying a specific trait, that I'll take. So I'll modify it as identify a specific trait. So you can also write what exactly you mean by this. So yes, we can identify different features. What is the gene composition for each feature? And if you want to create any special products, ठीक है, जैसे suppose एक plant आपने genome sequencing की, you found out there is this one sequence in this plant because of which the plant has special properties to live in less water. Okay, less water में भी grow करेगा because of that gene. That is available in some varieties. Now we can use this to create plant varieties by taking this gene, and plant varieties which will be using less water. Okay, we'll talk about that later on also, how we have the potential of taking or creating new varieties of organisms. अगर हमारे पास already information है of all the genes, we will be able to do that. यहाँ पे in terms of like COVID-19 variants we were talking, how do we know there is a new variant? Is if you have a data of what is the genome sequence of that virus. आप virus लोगे, उसका genome sequencing करोगे, then when a new virus comes, तो आपको check करना पड़ेगा, and then you can identify that okay, some part of its genetic material has changed, because of which we are calling it a new variant now. And the part which has changed, you can correlate it with the symptoms also. Okay, now that this part has changed, the mortality is less. Okay, this is less harmful because of this gene combination changing. So such information can be very, very helpful. Yeah. See, we'll talk about how we can create modular or what you call designer babies, right? But ये जो technology है, you have to understand that this is not for creating anything. Sequencing का मतलब है सिर्फ sequence लिखना, sequence understand करना, detail लिखना. What you can say is, based on this information, yes, we can create new organisms or modular babies or designer babies. We'll talk about how that is working. ठीक है, उसके लिए बेस इंफॉर्मेशन यही होगी. ठीक है. Yes, we can understand the lineage also from this. Like suppose we do a genome sequencing of people in India, people in Africa, people in Australia, right? The closer you are genetically linked to somebody, the more common your genetic material will be. And you check for commonalities. तीनों का genome sequencing कर लिया and you find that if we see the percentage of genes, our genes are more. Now this is a hypothetical example. So suppose our genes are more closely related, or our sequences are more closely related to Africa. So that will give an indication that yes, we have closer relationship in terms of ancestry from the African population than from the Australian population. You can also use this to understand links between different species. In terms of evolution, you will get a lot of understanding. जैसे human beings we know have evolved from monkeys, right? So अब monkeys में बहुत सारे monkeys होते हैं, different types. You have chimpanzee, you have gorilla, you have apes, right? So you can do a genome sequencing of all these varieties and then you can do one for human beings and check. जिसके साथ ज़्यादा percentage of genes match करेंगी वो अपना closest relative, right? Like suppose after from all of that you find that apes are the closest in terms of genetic link. So that will help us understand evolution also कि कैसे evolution हो रहा है. So that is why there's a lot of interest in doing this genome sequencing. India also has a project for this. So we have a very ambitious project where the idea is that 1000 Indians are going to be sequenced. Okay, when that will happen, let's see, because this is a very time consuming, very difficult exercise. But the idea here was that whatever genome sequencing projects were happening in the world, they did not have a lot of representation from India. Most of that was sequencing European genes. So because of that, India has come up with its own project. How DNA is damaged? So DNA डैमेज या चेंज का मतलब यही है कि अगर ये एक ही जगह टी आ जाए तो आप इसको बोलोगे डैमेज हो गया, ये चेंज हो गया. दिस इज व्हाट इज डैमेज. दिस डैमेज can happen through many factors, through some radiation exposures. जैसे you have had nuclear accidents in many parts of the world, like Chernobyl or Hiroshima and Nagasaki. So that kind of high radiation exposure was something which changed or damaged DNA. Even in smaller amount, they say X-rays can do this in the long term. Or naturally also in nature, the process is never exactly perfect. So there can be some alterations there as DNA goes from one generation to the other. Okay. Please write both these terms. First write genome. Genome. It is the complete set of DNA. It is a complete set of DNA of an organism. It is a complete set of DNA of an organism, which includes all the chromosomes, and the genes. Genome sequencing is describing the exact order of base pairs base pairs, nitrogenous base pairs in an individual, this data can be analyzed to understand the function of various genes, and identify changes. Diabetes is a genetic disease. Diabetes can be genetic, but it is not always genetic. So there are people who have diabetes right from the time of birth. So when that is happening, that is definitely genetic. That is definitely because of something to do with the genetic material and the genes. For most of the people, however, diabetes develops at a much later stage of life, usually due to some lifetime related challenge which they are facing. In that case, of course, we say that there is a strong hereditary factor there. If parents have diabetes, the chances of their child having diabetes when he grows older is higher. In this case also, this is not a definite correlation. अगर कोई चीज सिर्फ purely genetic है, then there is going to be a definite thing कि genes में defect है तो वो express होगा ही होगा. But even with that hereditary aspect, the person is at an increased risk, but if he takes good care of his lifestyle and takes timely precautions, he might never have diabetes. Okay? Okay. We'll take a break here, then we'll go further. Can you tell us about the viruses which is recently in news, like lumpy virus? No, I cannot. So you have to prepare these viruses from the point of diseases. So we did an overview about how viral diseases are happening or their basic features. But beyond that, we really don't have the scope to discuss all the viruses in news here in the class. Just see what disease they are causing, how they are causing, air, water, vector, what is their mode of transmission, and if there is anything specific about a government initiative at that end. That is something which you should be knowing. We are going to start talking about how inheritance is happening. We have been talking about how this genetic material is supposed to be functioning. It is needed for our characters, and characters are transferred from one generation to the next. So we are going to understand how that inheritance happens. To go into this, the first point which we are bringing in is the idea of chromosomes. That is the form in which DNA is present in our cells. And every cell or every person of a particular species has the same number of chromosomes. The number of chromosomes is fixed. If we look at human chromosomes, we find that we have 46 chromosomes, and these 46 chromosomes are present in each and every cell of our body of all the human beings. This is coming from our parents. The cells which are contributing to this is we spoke about this, the reproductive idea, yesterday also. So you have an egg. you have a sperm, the gametes, they meet, they fuse, and you get the first cell of life, which is the zygote. So life of an organism would be starting with a single cell, which is the zygote. This is the first cell of your life. In terms of chromosomes, if we have to think about this, like I just told you, all the cells are supposed to have 46 chromosomes. So if that is the case, egg में 46 होंगे, sperm में 46 होंगे, and अगर ये fuse करेंगे, join करेंगे, the number will become twice. So that means in every generation, this number will keep becoming double. So the idea of how a species is supposed to maintain 46, that is ruined. वो तो होगा नहीं, हर generation में number ही change हो जाएगा. So to solve this problem, nature has come up with a very smart solution. And that is that instead of 46, the egg has 23 chromosomes and the sperm also has 23. So that when they fuse, instead of 46, it's going to be twice of 23, which is going to be 46. In this way, every time reproduction takes place, every time chromosomes go into the next generation, this number 46 is going to be maintained at all times. This is fixed. Okay. We have standard terms to describe this. We have haploid and diploid. Haploid is represented as N, diploid is represented as 2N. So basically haploid is half of diploid. Okay, half the total number. Diploid is usually the total number of chromosomes of a species, and half of that is haploid. And this would be seen only in the gametes, like your eggs and sperms. So the gametes are haploid. They have n number of chromosomes. अगर human beings की बात करें तो n is 23. All the other cells of your body, other than eggs and sperms, the term for them is somatic cell. Your liver cell, pancreatic cell, muscle cell, all the cells of your body, they are somatic cells. They have 2n number of chromosomes. For human beings, this 2n is 46. For some other species, this can be something else. We are going to use this term n and 2n a lot of times. So you should be absolutely clear what this means. When you're writing n, you are representing a gamete. You're representing half the number of chromosomes of a species. When you're writing 2n, you are representing the total set of chromosomes of a species. You can see here in the nucleus, they have shown one pair or one set, and here they have shown two sets. And the contrast of these two sets, they have shown with two colors. Right? This blue and red color, this is supposed to represent कि एक जो है egg से आया है, एक part sperm से आया है. So every person, every human being has 46 chromosomes. 23 have come from the mother through the egg, and 23 have come from the father through the sperm. So if you think about this, there is no genetic or no biological basis of patriarchy. See, in terms of genetic contribution, both the parents are contributing equally. 23 chromosomes mother से आ रहे हैं, 23 father से आ रहे हैं. ठीक है? Let's write these terms. Please write haploid. Haploid. It is the single set of unpaired chromosomes. It is a single set of unpaired chromosomes represented as N. Represented as N. Gametes like eggs and sperms are haploid. Then diploid cell. It contains two sets of chromosomes. One from each parent. One coming from each parent. It is represented as 2n. Somatic cells that is all the cells other than the gametes, are diploid. Okay. Okay. We're going to go further to the next question, which is, if we are saying both parents are contributing 23 chromosomes, then how are these chromosomes coming, or which chromosome is coming from mother? What properties is it bringing? What properties is the chromosome of the father bringing? Right? That's the next question. So we have now sequenced all the chromosomes. Okay, we know what is the composition, what is the size, what is the shape of all the chromosomes, what gene they contain. That whole genome sequencing has been done. Okay. And if we look at our chromosomes, if you take one cell and arrange all the chromosomes, what we find is a picture like this. Okay, these are the 46 chromosomes of one cell of a human being. Can you see these are of different shapes and sizes? Yes, you can see the difference in shapes and sizes, right? Okay, so we have chromosomes of different shapes, different sizes, different structure. But what we find is that for each type, we have 2 chromosomes. जब हमने 46 chromosomes study किए, so we found that there are 2 of each kind. Like you have 1 and 2. 1 and 2. 1 and 2. ठीक है, उनको अरेंज भी इन्होंने पेयर्स में अलग अलग किया है. हर टाइप के 2 क्रोमोसोम्स थे, हर शेप साइज के 2 क्रोमोसोम्स थे. So what was further understood and studied was that these chromosomes are coming from each of the parents. We studied these different chromosomes, we gave them names. The names are in the form of numbers. They are numbered chromosomes. So इसको chromosome number 1 बोल रहे हैं, इसको 7 बोल रहे हैं, इसको 13 बोल रहे हैं. But for every type we have 2. We have 2 chromosome number 1. We have 2 chromosome number 7. We have 2 chromosome number 13. One has come from the mother and one has come from the father. Let me elaborate a little bit more on this. Egg से 23 chromosomes आएंगे, sperm से 23 आएंगे. So if we start checking the chromosomes of the mother, and we start arranging them. So we'll find 1, 2, 3, 4, 5, 6, so on till 23. If we further go looking into these chromosomes, so we might find, जैसे suppose, so hypothetical example, chromosome number 3 has the gene for eye color. Chromosome number 5 has the gene for hair color. Now if we start checking for the father, कि father के भी chromosomes लेकर we start arranging them, what we find is, again chromosomes, there is a chromosome which looks just like chromosome number 1 of mother, so that's also chromosome number 1, and similarly you have all the other chromosomes, same structure, same shape, same size. Further, when we go deeper into studying the genes, we find कि chromosome number 3 पे यहाँ पे भी eye color का gene है, chromosome number 5 पे hair color का gene है. That should give you an idea that for a person now there are 2 genes for eye color, there are 2 genes for hair color. There are similar chromosomes, they have similar information, they have similar properties. This should tell you that you have twice the information for every feature. आपके पास 2 genes हैं eye color के. Of course, within them, the eye color information can be different. Like the father can give you brown eyes color gene, ठीक है, जिसमें brown eye color बनाने का information है. The mother on the other hand can give you green eye color gene, right? उसमें अंदर content different हो सकता है, but both of those genes are for eye color. Ultimately, there is going to be an interaction between those 2 genes and it will be decided कि आपका eye color होगा क्या. Because I'm sure you know you will not have one brown eye and one green eye, right? Normally that will not be the case. So आपका eye color कुछ एक ही होगा. We'll go, that's the next step which we'll go to, that how that is decided. Here you need to know कि there is no exclusion. ऐसा नहीं है कि eye color will come only from father, hair color will come from mother only. That's not the case. ठीक है? If you think about this, if that was the case, then everybody's eyes would be like one parent. पर ऐसे नहीं होता. किसी की मम्मी जैसी होती है, किसी के पापा जैसी होती है, किसी की दोनों से नहीं मिलती. Right? That's also a possibility. That seems like a joke, but that can biologically happen. Right? We'll study how that happens. Okay. So, in this case, the point which you have to remember, or the point which I'm trying to make is, for every information, for every gene, you have twice the information because both parents have contributed to that gene. All the genes in your body, they are expressed twice. You have 2 genes for everything. Why you have 2 genes is because एक mother से आई है, एक father से आई. And because of this doubling of genetic material, we have 2 chromosomes of all these types. The genes are present on the chromosomes, and these pairs of chromosomes, they are called homologous chromosomes. So we can say that our 46 chromosomes are actually paired chromosomes and we have 23 pairs of chromosomes. Pairs क्यों? क्योंकि types 23 हैं और हर type के 2 हैं. ठीक है? Please write this term, homologous chromosomes. Homologous chromosomes. They are similar in shape. They are similar in shape, size. They are similar in shape, size, and gene position. And gene position. One coming from each parent. ठीक है? Okay. Now we go to the next question, which is brown eyes or green eyes. Okay. To understand that, we are going to take help from the experiment of a very crazy scientist called Gregor Mendel. This man, he's the father of genetics. Okay. And the brilliance of this man lies in the fact that he has worked at a time when there was no information of DNA, chromosome, gene, nothing of that sort. Nobody knew about that, but still he was able to study inheritance, and he has given laws which are still relevant, which work even now for us to understand how information is traveling from one generation to the next. Okay. So we are going to take a little help from the experiment which he conducted to understand inheritance of characters. He studied inheritance with the help of this plant, garden pea, your normal matar ka plant. Okay. Mendel was not working in any university or lab or anywhere. He was working out of his kitchen garden, growing matar in every generation and then predicting कि अब इसमें कौन सा feature आएगा next generation में. And there's so many stories written about Mendel that how he was, so anxious about his results, and if he wouldn't get the result which he was expecting, he would really beat up all his servants that you must have messed something up. कैसे हो रहा है, right? The success of Mendel's experiment actually lies in the choice of this plant also, because for garden pea, what he would observe was when he started checking for different characteristics or different features of this plant, he found there were only two variations. If he looked for height, so there was only tall plant or a dwarf plant. No medium height plant or any other variation was there. Sirf do extreme contrasting traits. If he looked at the color of the pod, he found pod is the outer covering of matar. Okay, कि या तो green है या yellow. If he looked at the flower, he found it would be violet or white. And just like this, he studied a couple of other features also. Now what he would do is he would take one tall plant, one dwarf plant, and keep reproducing multiple generations of that plant and see inheritance kaise ho raha hai. How is this feature of height being transferred from one generation to the other? And he was finally able to make calculations and probability diagrams, which we will also learn how we can make, and then we will apply to some of the human features. This is the image of one of Mendel's experiments. What he did here was, he took a plant with green pod and another one with yellow pod. Then upon fertilization, he found that in the next generation, all the plants were green. अब parents हैं, एक green है, एक yellow है, but all their kids are green. Then upon fertilization of this generation, he found again there is a yellow plant. Green तो है ही है, but yellow है. In this case, both the parents are green, but they have a yellow child, which was the trait of, you can say, the grandparent. It did not show in this generation, but somehow it has come up again in the next generation. So this he is going to now calculate that how this will work. So I am going to make probability diagrams for this. You are also going to make them with me, and you also are supposed to learn how to draw this, because they will be helpful for solving many different kinds of questions. The starting point here is from this idea. Here, this is where we'll start. When we studied about homologous chromosomes, we said that we have two genes for all our features. जैसे eye color की यहाँ पे 2 genes हैं, hair color की 2 genes हैं. So similarly, the 2 features with which we are starting, green and yellow pod. For this, for green color, the plant has 2 genes. Okay, just like eye color का 2 gene था, तो यहाँ पे भी green color के लिए 2 genes होंगे, and we are going to represent it as G. Okay, this representation has no significance. The gene does not look like this. ठीक है, so there are a lot of people who ask me कि मैम आपने small लिखा है कि छोटा लिखा है. आप जो मर्जी लिखो, जिस मर्जी भाषा में लिखो, उससे कोई फर्क नहीं पड़ता है. Just understand the concept. Yellow के लिए I am writing Y and Y. The yellow plant with 2 genes for yellow. Okay. The next thing we learned here was that for reproduction, special cells have to be prepared which should have half the genetic material of the overall species. So अगर species में 46 है, reproductive cell में 23 होंगे, half. Same rule we'll apply here. Here, if the organism's composition, because we are considering only one feature, तो यहाँ पे अगर 2 हैं, gametes की stage पे या while preparing for reproduction, this has to separate. So 2 की जगह it's going to become half, and you will have G and G. Similarly, यहाँ पे Y and Y. Now, if these are the gametes, they have to fertilize. That means they will combine. One gamete from one parent, one gamete from the other parent. But क्योंकि यहाँ पे multiple gametes के options हैं, we have to check all combinations. This G can join to the first Y. G can join to the second Y. Second G, first Y. Second G, second Y. On observation, Mendel found that they are all green. In appearance, they are all green. Now, if I take 2 of these, the plants which have come up in this generation, which will be GY and GY, I can repeat this cross with these 2 organisms, these 2 plants. There was a student who asked me कि ये भाई बहनों का आप reproduction क्यों करवा रहे हो? Right? So biologically there is no impediment to that. It can be done. Okay. And these are plants. So we made the gametes, and now we are going to check for all combinations again. G and G. G and Y. आप YG लिखो या GY लिखो, it means the same thing. It's just the combination which has significance. So I will write GY only, just for the sake of uniformity. And the last case is going to be Y and Y. Now from the first cross, we know that GY and GG both are green, but YY is yellow. Now this coming of a yellow plant in the 3rd generation made Mendel think that whatever is causing yellow cannot really jump a generation and reach here. It must be present somewhere in this generation. यहाँ पे उसका impact नहीं है, but it must be present. And he further concluded that it is present, but it is not having its impact. Because the plant is green. ऐसा नहीं कि G और Y मिलके कुछ नया color orange बन गया. It's not going to happen. It's either green or yellow. Only 2 variations. So Mendel gave this idea that out of these 2 features, one is dominant and one is recessive. In this case, when GY are coming together, the plant looks green. That means green or yellow दोनों की genes थे, but only green's impact is seen. That's why the plant is green. Yellow has no impact here. So green is dominant, yellow is recessive. In nature, what is dominant, what is recessive, that is fixed. That's not something which we can predict or something which we can change or alter. ये fixed रहेगा. Whenever there is garden P and you're considering color of pod, in that case, green color will dominate, yellow will be recessive. Every time it's going to be like this. How we know what is dominant, what is recessive is when you see a combination like this. If one gene is for green, one is for yellow and the plant is green, that means green is dominant. अगर GY yellow होता तो you could have said yellow is dominant. ठीक है? Okay, let's write a little bit. Then we'll you introduce some more terms in this. Mendel को gene नहीं पता था. The term Mendel has used is factors. Okay, so we'll write this. All the characters in an organism are represented in two factors. These factors or genes are the units of inheritance. These factors or genes are the units of inheritance. The dominant trait is that inherited characteristic. The dominant trait is that inherited characteristic which overrides the impact of the other trait. The recessive trait is that inherited characteristic which cannot express itself in the presence of the dominant trait. We'll write this example also here just so that you can remember. For example, in garden P, the green color of pod is dominant over the yellow color of pod which is recessive. Now we'll be writing a few terms. Please write down. Homozygous Write this term. Homozygous is when both the genes are the same. Homozygous is when both the genes are same. So example of combination will be GG and YY. Dono genes for the same feature. That state we call homozygous. So this is homozygous, this is homozygous. Second term is heterozygous. Heterozygous when both the genes are different. Heterozygous when both the genes are different. Like in GY. One gene for green, one for yellow. The next term is phenotype. The next term is phenotype. Phenotype is the observable characteristic. Phenotype is the observable characteristic, like height, color, etc. Like height, color, etc. Then you have genotype. Genotype is the genetic composition. Genotype is the genetic composition. So basically this means कि GG है या GY है या YY है. This will be the genotype. If for this particular cross, this one, I ask you, what will be the ratio of phenotypes? There are two phenotypes here, two colors, green and yellow. So what will be the ratio of green to yellow? It will be 3:1. And if I ask you the genotype ratio for this cross? 1 is homozygous green, both genes for green. So there's just one of this. So we write 1. GY, you have two of these, GY and GY. So 2 for this heterozygous form. And for the yellow gene, when you have two yellow genes, that's just one of them. So it will be 1 for this. So 1:2:1. You can also see this कि जो trait recessive होता है, it will only express itself in homozygous form. The dominant one will express in both the cases. चाहे homozygous है, चाहे heterozygous form है, dominant will always express itself. But the recessive one will only express once it is in the homozygous form. Is this part clear? You understand what happened here? Okay. Now, please repeat genotype ratio. So, genotype ratio मैंने बनाया है GG ratio GY ratio YY. The combination of the genes. So, you just have one of these GG, two of GY and one of YY. I've just picked it up from here. Now, Mendel's laws or Mendel's inheritance, we can apply that for human features also, but not all features. Why not all features? Because we have a very complex inheritance process where for one feature multiple genes might be involved. जैसे आपका actual eye color बनाने के लिए there might be like 5 genes which will be contributing some part, some step, and then that final eye color will be reflected. So not everything we can predict, But we have certain features which exactly follow Mendel's inheritance. Okay, so yes, we will be talking about one such feature, which is your blood group inheritance. And I hope you find this interesting, because after studying this, you will be in a position to make your own inheritance cross. If you know the blood group of your parents and your own blood group, so you can do that whole thing for yourself. In blood group inheritance, so we studied yesterday that we have two types, ABO and RH type, right? RH ऐसे predict नहीं कर सकते हो, but ABO can be predicted. And that's what we are going to learn, that how ABO blood group inheritance is going to work. So you have phenotypes. The phenotypes here are the different blood groups which you have. So A, B, O, AB, 4 phenotypes, 4 variations. And we will look for what is the gene combination responsible for these blood groups or these genotypes. Just like for green color, we represented it as G and G. For A blood group, we are saying the genotype or the gene combination is A and A. We are calling the gene A. So A and A will give you A blood group. Similarly, we are saying the B blood group gene is B. So B and B will give you B blood group. O के लिए we are saying the gene is O. So O and O will give us O blood group. Now we just observed कि homozygous के अलावा heterozygous forms भी हो सकती हैं. Like we had something like GY. Here the heterozygous forms which we have could be a combination of these genes. A we find is dominant over O. The A gene is dominant over O gene. So when A and O is together, the blood group will still be A. जैसे G और Y था, पर it was green only. ठीक है ना? No impact of the Y gene. Same goes on here. A, O being together, blood group is A. B is also dominant over O. So B, O being together is blood group B. ठीक है? A dominates over O, B dominates over O. So that should tell you that O, because it is recessive to both A and B, इसका और कोई genotype नहीं हो सकता. Right? The recessive one only expresses itself in a homozygous state. The last one, AB, this is a deviation from the classical Mendel's rules, where when you have A and B together, they do not have a dominant recessive relationship amongst themselves. They both dominate over O, but when you have A and B genes together, they are what we call co-dominant. That means they both express themselves. And because both genes express themselves, a new blood group is formed, which is AB. They do not have a dominant recessive relationship, which leads to creation of another blood group type, which is AB. When both A gene and B gene, they both give their impact. ठीक है? Okay. Inheritance pattern, the crosses which you will need, they're going to be exactly like the Mendel's cross, and we have one question based on this. So UPSC asked this question, and this kind of question is a very, very common pattern. So read the question, make a cross, and tell me the answer. D. Okay. So if you have the answer, now you can solve for your own inheritance pattern also, if you know your parents and your blood group. Any answer other than D? A. Okay. People online, any answers? Okay, Vishakha says it should be A. It's a lot of A's I'm getting. जो D वाले हैं वो परेशान हो रहे कि कैसे आएगा इनका आंसर. चलो देखते हैं. A married couple adopted a male child. A few years later, twin boys were born to them. So there is a couple who has adopted one child and has twins. So total 3 children. Okay, 2 parents, 3 children. The blood group of the couple is A positive, O negative, then A positive, B positive, and O positive is the blood group of the 3 sons. And they are asking you the blood group of the adopted son is. So जो भी 3 blood groups हैं वो दिए, and the fourth option is cannot be determined on the basis of data given below or above. So this option is obviously given because you don't know how to solve for RH factor. Okay, so you read about RH factor and then you immediately go and mark this D. But whenever you have such an option which says cannot be determined, तो जितना कुछ आता है, based on that you must give an attempt. Okay, so you don't know how to solve for RH, but you know how to solve for ABO types. So let's just pick up ABO type and see what is happening here. So you have 2 people, the parents AB and O. पॉजिटिव नेगेटिव का हम कुछ नहीं कर सकते, तो हम उसके बिना ट्राई करेंगे एक बार. So the 2 parents are AB. AB genotype is also AB, right? So gene combination A होगा. O के लिए genotype will be O and O. Okay, so these are the genotypes of the 2 parents. We will make a cross now. So when we make gametes, it will be A, B, और यहाँ पे both will be same O and O. Now we are going to check for all possible combinations when they will have children. So A can join to O, A can join to the second O, B can join with O, or B can join with the second. This is all the possibilities of all the children which they could ever have. Okay, AO, which blood group will this genotype give? A. ठीक है, AO is blood group A. BO is blood group B. This cross tells us that when these 2 people have children, their child can either have blood group A or B. There is no other possibility. या A blood group हो सकता है या B हो सकता है और कुछ नहीं हो सकता. हमने सब तरफ से ट्राई कर लिया. Now let's see what the information is there in the question. It has already told you what are the blood groups of the 3 children. So the blood group of the 3 children are A, B and O. This is already given. Okay, इसी में से choose करना है हमें that which is the adopted son. Now we have calculated and seen that biologically these two people can only have a child either with blood group A or B. O they cannot produce. So blood group is actually one of the most basic paternity tests there is. Of course ये बिल्कुल हर केस में work नहीं करेगा, but in this case it's working wonderfully. O they cannot be their child biologically. That means if they have a child with O blood group, that child must be adopted. So the answer would be A, O positive. This question is misleading in the sense of representation of RH, and for that you have option D also to mislead you to the wrong answer, but बिना RH के भी easily solvable. ठीक है? Is this clear? Any confusion here? Okay. So you can check for yourself. I'll tell you my cross. Okay. So both my parents, they are blood group A and my blood group is O. So because my blood group is O, my genotype is absolutely clear. A में challenge ही होता है, A में AA भी हो सकता है, AO भी हो सकता है, you can't predict. But because of my blood group, now it is obvious that both of them were heterozygous and they both gave me their O, so I have O blood group. If the question will be B and A of a couple, then there will be 4 genotypes. How to solve them? So again the question will be given in a way that it will be solvable. If you have A and B, yes, you are very right. आप नहीं solve कर सकते क्योंकि दो genotypes हैं, इसीलिए उन्होंने A, B और O दिया है, ठीक है, ताकि कोई variation ना हो. They would have given you some information to solve. You cannot assume AA or AO. That's not an assumption which you can make in any case. So either it is given to you in some form, like maybe they say कि this is homozygous A or this is heterozygous A, or maybe the blood group of a grandparent is given, right? तो उससे आप predict कर सकते हो, otherwise there is no point of assumption here. हाँ, फिर option D भी है ये. Yes, that's right. Then option D will work. Good. Okay. So I always give this one example also in the class. So I have a friend whose blood group is AB, and she says one of her parents is A and the second parent is O. Okay. So my understanding here is this, that there are a lot of people who don't know their blood groups. Okay. Or suppose, you know, they got a test done some 15 years ago and then they've forgotten about it. तो वो AB का सिर्फ A रह गया, right? या B का A रह गया, कुछ ऐसा हो जाता है. So I keep telling her that, you know, all 3 of you must go one day and get your blood group checked. Because where does this come from? Right? But then she doesn't understand. She doesn't want to understand. She finds it very funny कि तू पागल है. I was like, okay, no problem. So check for yourself, okay? The next thing which we are going to talk about is sex determination. My mother is O positive, father is B positive, brother is O positive. So if B is having dominance over O, then why O has come out as a result? I didn't understand. अरे मेरा भी तो O है, वही तो समझाया आपको. When you form gametes, so they will individually be expressing themselves. ये देखो, dominant and recessive works in a combination, but in gametes it will individually go. जैसे if you say that your mother is O, तो mother का तो clear है genotype, ठीक है. Father is B and your brother is again O, right? So इससे father का genotype is also clear, right? Father will be BO. Okay. Now the question which Vishakha has asked is कि अगर B dominant है तो O क्यों आ रहा है? Dominant recessive works when both the genes are together, but when gametes will be formed, ये दोनों अलग अलग हो जाएंगी. एक gamete में B आ जाएगा, एक में O आ जाएगा. यहाँ पे चलो सब जगह O ही रहेगा. These are separate gametes. We said that the eggs or the sperms, they were supposed to have only half of the chromosome. So only one gene out of the two genes they will have. So जब ये B बन रहा है, ये अलग है, ये अकेला है. O जो है वो अकेला है. These will be in different gametes. So if you're talking about your father, so these will be two different sperms. यहाँ पे इनकी कोई इंटरेक्शन नहीं है. ये जब कॉम्बिनेशन बनाएंगे आगे जाके, then this combination will be where the dominant recessive thing will work. But otherwise, both the genes can be equally inherited. They are independently inherited with no relationship of with the other. ऐसा नहीं है कि B dominant है तो B कहेगा मैं ही जाऊंगा, तू यहीं पे रह. ऐसा नहीं होगा. Both the genes can be individually inherited. ठीक है. So for your brother, your father gave that gamete जिसमें O वाला जीन था, mother के पास तो वही है. So वो combination will work. Please. Then हाँ, only B blood group. अगर both parents are, see, homozygous and heterozygous, somebody has asked. जब दोनों जीन सेम होती हैं तो उसको बोल दिया होमोजाइगस. B है होमोजाइगस. अगर एक जीन B के लिए है, एक O के लिए, ये हो गया हेट्रोजाइगस. When both the genes are different. A है होमोजाइगस. AB एक जीन A का एक B का, दिस इज हेट्रोजाइगस. ठीक है. ओके. Coming to sex determination, that what is going to be the biological sex? Will it be a male? Will it be a female? When we look at our chromosomes, the image which we saw of the 46 chromosomes, this one. In this, we said we have 23 pairs of chromosomes. Out of these 23 pairs, there is one pair which is a pair of sex chromosome. In this, you have two sex chromosomes. One is called X, the other is called Y. You can see these two, X and Y. They are slightly different also in shape and size. The combination of these two chromosomes decides the sex of a person. अगर XY है, the person will be a male. If it is X and X, the person will be a female. It is this last pair of sex chromosomes which decides the sex of a person. जो बाकी chromosomes हैं, the rest of the 22 pairs, we have a term to describe them also. It is called autosomes. Let's see this again. So we have 22 pairs . So we have total 23 pairs of chromosomes, right? Which is comes to your number 46. Out of these 23 pairs, 22 pairs are called autosomes. And there is one pair which is called the pair of sex chromosomes. In this, we have two sex chromosomes, X and Y, and the combination decides the sex. Agar X और X मिल जाए, it becomes a female. That means female reproductive organs, female reproductive cells, eggs will be present. And if it is XY, so it's going to become a male. Male reproductive organs, male hormones, male sex cells or gametes. If you think of this in terms of how this is transmitted, if I take you back for this image, in the normal cells or in all somatic cells, you have 46 chromosomes, but we have studied that for gametes, that is your eggs and sperm, which are going to go into the next generation, in that this number will be half. तो ये पेयरिंग टूट जाएगी और हर पेयर में से एक एक क्रोमोसोम जाएगा. ठीक है, ये पेयर्ड है, बट in gametes instead of 46 you have only 23. So one of each type will go into the gamete. If I have to write this down, how I'm going to put this? So for the mother or for the female, she has total 46 chromosomes, but when she will form the egg, she will only give 23 chromosomes. Yes? अब ये 23 में से 22 तो ऑटोसोम्स हैं, ठीक है, 22 पेयर्स में से एक एक आ जाएगा, and then the last pair, she has both X chromosomes, so one X will come. Her egg will have 22 autosomes plus one X chromosome, total 23 chromosomes. ठीक है. In case of father also has 46 chromosomes, but the sperm is supposed to have only 23. 23 में 22 autosomes will be there, but for the sex chromosome there is a variation. इसमें दो अलग अलग chromosomes हैं, so that would mean you will have two types of sperms. So एक में 22 plus X, the other possibility is 22 plus Y. Which one will come and fertilize the egg? That will decide the sex of the child. This also follows the simple Mendel's inheritance. So you can draw that cross also. This is the father's genotype X and Y. अगर gametes बनाने हैं, so we can make X and Y, two kinds of gametes. Mother में X and X, both gametes are the same. So you get XX, XY, XX, XY. This is a natural process, selection is automatic, but if I ask you what is the ratio of phenotype here, What will that be? Phenotype is the character you're observing, and here you're observing the sex. So you have daughter and son, two phenotypes. What is their ratio? 2:2 or 1:1. Same way, what will be the genotype ratio? 1:1. जो जीनोटाइप है वो भी कॉरस्पॉन्ड कर रहा है phenotype से. So you have XX and XY. So 2XX, 2XY, 2:2 or 1:1. Again, same thing. Probability is 50%. That is why in nature, when you look at a population, the sex ratio is close to 50%. In the ideal scenario, it's supposed to be close to 50% because of the way natural probability will work. However, if you have to look for what is determining it, it is going to be which sperm is going to come and fertilize the egg. You know, in our country, we have this problem of preference of male child, right? And the burden of giving birth to a male child is on the female. You know, how people will accuse the female that, you know, this woman, she's not going to give us the heir to our throne, Vansh ka chirag and all those things, no? So if you could explain this to them, it will solve so many of our problems, right? See, the mother has only X chromosome. Her eggs have no choice. Okay. You cannot actually blame anybody because this is happening naturally, right? But if you still want to blame the sperm. Okay. So there are two types of sperms here. One will have X chromosome, one will have Y. Agar X wala aake fertilize karega, you'll get a daughter. If the Y one will come and fertilize, it's going to be giving a son. Of course, people are going to hit you once you try to explain them this, but you can still try. What could be the transgender chromosome? Okay, so there's nothing like a transgender chromosome. First thing here which you need to understand is gender is not a term which we can use biologically. The term which we use is sex. Okay, biological sex. If you paid attention, that's the only word which I've used. Gender is a social construct. Is the matter of a social identity which is assigned to a biological sex. When I say biological sex, I mean the reproductive system, the organs, presence of ovaries or testes, presence of hormones, all those things. Okay, that is something which is determined by these chromosomes and the hormones which are there or anything like that. When you say gender, that becomes a broader term. That is all those properties which the society has assigned to being a woman or to being a man. Okay, something like, you know, women are supposed to dress up in a certain way, talk in a certain way, have one kind of job. That's the gender identity of being a female or being a man in that sense. When you talk about this term transgender, it is a very, very big concept and not just limited to the sexual identity. There is a possibility कि इसमें भी problems हो सकती हैं. We're going to talk about and study that also in our next class. That how you can have some alterations, you can have some extra chromosomes, some missing chromosomes, जिनकी वजह से the person will be in a situation where he's not going to be exactly or completely reproductively active or complete functioning. जो usually आपके eunuchs जो होते हैं, ठीक है, they are the people who might have some of these chromosomal abnormalities. But that number, that ratio, that proportion is very, very small. Presently, the concept of gender is or transgender identity is very broad. Okay, there can be some people who can have some hormonal issues also. ठीक है कि you are a female with XX chromosome, but you have very high amount of male hormones, or a male has very high amount of female hormones, which can create certain variations. But beyond this, there is a very big scope of interpretation of this term of gender, right? Like if I have to give you examples in terms of gender identities, you might have seen some movies or some documentaries or something where people say that I was born as a boy but I never felt like a boy, right? You know those stories, right? And then there are people who will further talk that why I never felt like a boy is because I like to wear lipstick and heels, I wanted to dress up in my mom's sarees and things like that. ठीक है? अब ये सब चीजें biological नहीं है, right? The body was not supposed to, was not created to wear lipstick or heels or anything like that, right? That's not determined by your chromosomes. Otherwise, your legs would have been in a very different shape if you were born to wear heels, right? But that's not the case. This is again a societal idea. The idea of how females are only supposed to wear heels and men are not. This, when people contradict to these ideas, or when they talk about becoming transgenders or not liking the gender or the sex which they have at birth, that is mostly a psychological preference. But uska koi biological basis nahi hai. You really don't talk about anything being wrong in this structure. This might be working perfectly, but psychologically they are not aligning, or they don't want to align with the societal description of what a female should be or what a male should be, right? So in that case, there are people who wish to get a surgery also done, right? Something which is called a sex change operation. It's not actually sex change क्योंकि ये change नहीं हो सकता. At least अभी जो technology, you can't change this. Whatever chromosomes a person is born with, that decides everything, that decides the internal organs in the body, that you cannot change. People who have a sex change operation, they just cosmetically change the external appearance of their body, and then they take hormones to maintain that. Just say if a man is to become a female, he will need female hormones, right? So that his hair is like that, his voice is like that, all of that he'll need to maintain for the rest of his life. लेकिन टेस्टिस निकाल के ओवरी डालने का अभी कोई प्रोसीजर नहीं है दुनिया में. So that will not happen. Only the external body can be changed. ठीक है? Again, there is no biological problem here. Okay, but it is a matter of inclination. Now this concept has expanded to so many things, like there are people who don't want to become a part of either groups. कि हमको मेल भी नहीं बनना है, हमको फीमेल भी नहीं बनना है. So there's this idea of non-binary concept. So there people, in many countries in the world now, they've actually started asking children that what pronoun should we use for you? You want us to call you he, she, and then there are many other variations, it, they, them, whatever. A person can choose, right? कि क्यों मैं ही क्यों बनूं, मैं she क्यों बनूं, क्या इसका आइडिया है मेल फीमेल का? तो मुझे कुछ भी नहीं बनना है. I want to be non-binary. So that's also there now. All of that will come under this transgender or other gender identities. Most of these things are matters of preference. Okay? There is also an angle here of sexuality or sexual preferences, right? When you talk about LGBTQAI plus, abhi they say there are 70 plus categories in this, whatever that is. There is a big section where when you talk about homosexuality or asexual or bisexual, whatever that is, again, that does not have any kind of biological basis in terms of something wrong with their hormones or chromosomes or anything like that. ऐसा कुछ नहीं. Everything is perfectly fine. It's just that they want to make a choice of whom they want to have a sexual relationship with. That's it. So this is a topic which you are going to come across, especially now with the Supreme Court and everything involved in same-sex marriages and all of these ideas. As civil servants, it's very important that you look at this thing with a lot of objectivity and you are very sensitive to this whole matter. Because most of the people involved here, they are trying to express a choice here. right? This is not a disease or a problem or anything like that, right? But this is a matter of right. As the discussion is going, this is my right, whom I want to get married to, whom I want to have a relationship, or how I want to dress up, or if I want to call myself a female or a male or whatever that is. So in your approach, when you are analyzing this issue or when you are looking at it, if you have to talk about any challenges or any issues here, those have to be objective challenges. Objective in the sense ki, okay, if we have to do this, what kind of variation do we need in our acts? ठीक है, हमारा हिंदू मैरिज एक्ट है, अगर हमको ये मैरिज अलाउ करना है तो उसमें किस टाइप के चेंजेस चाहिए, क्या चैलेंज आएंगे, इंफ्रास्ट्रक्चर के मैटर पे क्या किस तरह के चैलेंजेस आएंगे. ऑल दोज थिंग्स आर व्हाट यू कैन ऑब्जेक्टिवली थिंक अबाउट, बट बायोलॉजिकली एवरीथिंग इज फाइन. कुछ लोग हैं, there is a section which will have some problem with their chromosomes and their biological sex for that matter, their reproductive system for that matter, but other than that there is nothing scientifically which you can link as a problem in this issue. ठीक है? How many classes are left? Oh, somebody got bored with my discussion. So let's see. One for sure, maybe two. देखते हैं how it goes in our next class. So we're going to end here. And in the next class, we'll be continuing this topic only. So there is this class 10 chapter on reproduction and heredity. We've not studied a lot of it in heredity, but just if you want to refer, we'll be doing most of it in the next class. You can check this out if you need. Okay. Thank you. Have a nice day.